Well, welcome everyone. So glad you could join us. This is Doug McConnell. I'm a co-founder of a nonprofit called A Long Swim that uses open water and marathon distance swimming to raise money and awareness for ALS, Lou Gehrig's disease. The laboratory that we most closely embrace and support is the Ozdinler lab at Northwestern University's Feinberg School of Medicine here in Chicago. And one of the real reasons behind our dedication to the Ozdinler lab is the lab's real commitment and dedication to collaboration and research with other ALS centers around the world, and even within the four walls of Northwestern itself. In that regard, today we have a very special guest, Dr. Richard Silverman, whom I've had the pleasure to get to know. Dr. Silverman is I'm going to read this so I get it right, Rick, the Patrick G. Ryan and Aon Professor of Chemistry at Northwestern. He's been with Northwestern since the mid-1970s. And really has dedicated his career to drug development and testing, particularly associated and focused on central nervous system disorders, including things like ALS, of course, but also Parkinson's, epilepsy, and others. One very important claim to fame that Dr. Silverman has is really being the father of Lyrica. I hope I'm saying that appropriately, Rick, but Lyrica is one of those things that you can't listen to the evening news and not learn about Lyrica. It has turned out to be a wildly successful drug. And I think that important to this conversation is how Dr. Silverman and Dr. Ozdinler's labs discover compounds in the first place, and once they show promise, really escort them through the process to ultimately become pharmaceuticals. So, Rick, I'm so happy to hand this over to you and to talk a little bit about NU-9, which is a recent discovery that has come out of your collective laboratory.
So a number of years ago, we were thinking about some kind of new treatment for ALS. And for decades, there have been drugs tested in animals, and they seem to be effective in animals through a test that was, let's see if the drug extends the life of a mouse that has the same mutation that a small percentage of ALS patients has. But none of those drugs actually turned out to be effective in humans. And so we were trying to think, well, what can we do, take a different approach? And we noticed that ALS, as well as all neurodegenerative diseases, have one thing in common, and that is that they aggregate proteins, different proteins for different diseases, different parts of the neuron, but they aggregate proteins, and that apparently is causing the effect of that disease. So we thought we would look for molecules that inhibited protein aggregation. And at the time we were interested, as I say, in ALS. And the mouse model that was used for ALS is one in which there's a mutation in a gene that occurs in about 2% of ALS patients. And that then produces this aggregation of that particular protein that comes from that gene. So we set up an assay to look for molecules that would inhibit protein aggregation as a result of the mutation in that gene. And we screened randomly over 50,000 compounds. And we found that there were 17 different structures that had an effect on this protein aggregation. And so as a chemist, I could look at these structures and see which ones would be maybe simplest to synthesize, that have proper pharmacokinetic properties so that it would be transported well, absorbed into the body. And I came up with 3 different classes of compounds. And so we focused on those 3 that were active in this assay, and that we knew we could get patents on them if we needed. And we started to develop them. And as a chemist then, once we have a starting point, we can make changes to the structure of the molecules and then retest it to see if we improve its ability to inhibit protein aggregation. And we did that with all 3 classes of molecules. And after several years of doing that, we came up with 3 optimized molecules. So then we went to the next step to see, well, which of these gets into the brain best, because you need to do that if you want to have an effect on ALS. Which ones are stable in the body longest? Which ones don't have off-target effects and might cause side effects? And so after doing those studies, we settled in on one of those 3 classes, and the best of that class of molecules we then called NU-9. And we tested it then in a variety of different studies, including looking at its effect on the lifetime of this ALS mouse. And it did extend the lifetime of the ALS mouse, but as I said before, that doesn't mean that that's going to have any effect in humans. I then got connected through Northwestern with Hande Ozdinler, who at that time had just come from Harvard to be at Northwestern Medical School. And we started to talk about this. And she had I felt, a very intriguing idea based on her research, that the lifetime of the mouse really doesn't matter. What matters is the health of those motor neurons that are going to cause ALS. And if your molecules, my molecules then, would stabilize and keep those motor neurons, and these are upper motor neurons, the corticospinal motor neurons, if we could keep those healthy with my molecules, then there's a good chance it would translate to humans, because those neurons in mice and humans are really quite similar. So that's how we got connected. And Dr. Ozdinler can tell you what she then did with us.
Wow, that was very good. Thank you so much, Rick. Yes, I remember the day, we met and then we started talking, and I think our collaboration got better by the day, by the year, and hopefully we will have even more discoveries to make. So I would like to share my screen if you don't mind. Oh, now host disabled participant screen sharing. So maybe if you can allow me to share my screen, that'd be nice, because I would like to show you how we allowed or developed ways to visualize these upper motor neurons, and how we were able to develop cell-based and mechanism-focused applications. The goal of today's meeting is to talk a little bit about this new publication that we have, and how NU-9 actually improves the health of mitochondria and ER, and how that helps eliminate upper motor neuron degeneration that occurs due to SOD1 toxicity and TDP pathology. So first, I need to tell you that the upper motor neurons are actually critically important for ALS, because the movement starts in the brain. And for the very long time, the cortical component or the brain component of motor neuron circuitry has been a bit ignored, because the spinal motor neurons connect to the muscle, and the focus was improving the health of the spinal motor neurons. But actually, the spinal motor neurons receive input from the brain, from the upper motor neurons from the corticospinal motor neurons. Therefore, if we have a very healthy brain, eventually and subsequently, the spinal motor neurons would also improve health. But then how do you study the upper motor neurons? Because the brain is so complex, right? So we actually made a reporter line in which the upper motor neurons are fluorescent. Can you imagine? Not all neurons in the brain are fluorescent, but just the upper motor neurons are fluorescent. And maybe I can show the slide here. And you know, they become green and fluorescent, which allows us to visualize them, and which allow us to assess their cellular requirements for survival and for drug discovery. And you can then imagine that there are so many different disease models that are developed based on the mutations detected in patients or pathologies detected in central nervous system of patients. We can cross them with this reporter line, and now we have diseased upper motor neurons that are GFP that we can visualize. And of course, with this ability, we were invited by Drug Discovery Today a couple times, and together also with Dr. Silverman, to write review articles on how we can move forward the clinical trials for ALS by asking motor neurons what they need, what they want. So as Dr. Silverman said, improving the health of the mouse does not really translate to improving the health of the patient. We have to improve the health of the diseased neurons. So this gave us an ability or a chance to assay compounds for their ability to improve the health of diseased neurons at a cellular level. Then we made a very important discovery. We realized that the underlying pathologies or underlying problems of upper motor neurons in humans versus mouse, even though they are different species, are exactly the same. So then, even though there's no translation from mice to human, there is translation from neuron to neuron. There's translation at a cellular level. So for example, one of the major problems was in the mitochondria. They were broken, especially the inner membrane. And when you have mitochondria like this, you can't really produce ATP. You need the intact inner membrane to produce ATP. Then we realized that in the mouse, it's also the same problem. The inner membrane have holes, the mitochondria is falling apart. And the ER normally is like this in a book, it's stacked, and then you see the ribosomes. But in the patients, in their upper motor neurons, beta cells, these ER is enlarged or they are falling apart. And we see similar problems with the upper motor neurons in the mouse as well, and in different two models, both in the SOD and TDP. For TDP, I would like to say that's actually one of the most common pathologies in patients. About 80–90% of ALS patients, especially ALS FTLD patients, have TDP pathology. So if we can actually improve the TDP pathology, that would have a broad implication for many ALS patients. So once we realize that there are common causes of neuronal vulnerability, we then administered a NU-9, as Dr. Silverman has said, by oral gavage every day for 60 days in two different doses, 20 milligram per kilogram per day or 100 milligram per kilogram per day. So a mid dose, a bit high dose. Then we looked at the mitochondria ER. Then we said, okay, NU-9, can you improve the health of mitochondria? Can you improve the health of ER when, in those upper motor neurons when we begin to give them at P60? That is the time that they show symptoms. So that would be very similar to a patient developing ALS symptoms. And lo and behold, you will see the details in the paper. Yes, the mitochondria improves, ER also improves. Here are the quantifications, statistics, and you can read more in the paper as well. Then we said, okay, so the mitochondria in ER is improving, but does that translate to improvement of neuron health and retention of neurons in the motor cortex? Because as we know, with disease progression, these neurons progressively degenerate. So by giving NU-9, can we actually keep neurons in the brain and can we stop their degeneration, right? So in the wild type, you can see beautiful upper motor neurons. These are all GFP. And in the SOD1 mice, you can see that there are very few left. They are sparse and they are small. And NU-9 treatment does something, but 100 milligrams is actually almost like the wild type levels. And each dot here is like a mouse. So you can see that its N number is pretty high. And we have seen that with the addition of NU-9, with treatment of NU-9 for about 60 days, the levels were almost similar to wild type levels. So which means that we can actually stop the progression of upper motor neuron degeneration, and that may be due to improved health of their mitochondria and improved health of their ER. The other thing that we looked, and that's very important, I would like to emphasize, we looked at the stability of the apical dendrites. And you may wonder, what is an apical dendrite? So apical dendrite is the extension of this neuron all the way to the top layers of the cortex. So this apical dendrite is very important for cortical connectivity. Because upper motor neurons carry information from brain to spinal cord, they need to be modulated by other cortical cells and other cortical neurons. And how are they modulated? They are modulated by the connections that they receive in this very important site that we call apical dendrite. So in a healthy neuron, the apical dendrite looks like this, okay? And there are even spines. And in the diseased neuron, the apical dendrite looks like this. They don't have connections, they have vacuoles, they're falling apart. So when you have an apical dendrite like this in an upper motor neuron, it basically means that they can't really receive too much information, and they can't really convey that information to the spinal cord. And that's one of the reasons why there's a disconnect from the brain and the spinal cord. So we saw that if you actually have NU-9, right, here are the wild type levels with about 100, this is very similar to the wild type levels. So then the apical dendrite also regains its stability. Look at this, right? It varies very similar to the wild type. So then NU-9 not only improves mitochondrial health and ER stability, and it also improves the cytoarchitectural dynamics. So then those 3 points are very important for motor neuron disease. And here, this was a slide showing why upper motor neuron apical dendrites are important, because this is the site where all the connections are made. And if this degenerates, like as we see in other disease cells, in disease neurons, they cannot receive input. So we have to make sure that the apical dendrite is intact. And we actually found that this apical dendrite defect is not a mouse phenomena. You also see this in familial ALS cases, in FTD ALS cases. Look at their apical dendrites. They're filled with vacuoles. But you don't see this in the Alzheimer's disease patients, because in the Alzheimer's disease patients, the problem is not the motor neuron circuitry, it's hippocampus, right? And it's very neuron specific, and it is very disease specific. So our goal is actually to make them look like the normal controls. And
Hande, can I interrupt just for a second?
Of course, of course.
One of the questions that seems to keep coming up in some of the articles, excuse me, that have been published about NU-9 is this issue of whether it slows the deterioration, or in some cases actually reverses the deterioration and repairs those upper motor neurons. And I think your description of these dendrites and so forth is terrific. But how is that the process by which we measure whether the deterioration is arrested or improved?
Okay, this is a very important question, because we begin to give NU-9 at P60, and that is the time that the upper motor neurons are vulnerable. Okay, so there are stages of degeneration, right? First, they raise a red flag and they say there's a problem, and then they become vulnerable, which means they live with that problem, and they say, I don't like this, they suffer. And then after a while they say, okay, I can't do this anymore, and they initiate apoptosis, and there's a neuronal death and neuronal degeneration. Then astrogliosis, microgliosis come in effect, and they chew them up, they clear, and then no more neurons left in the system. So there are stages, right? So we give them at a time when they are still unhappy, but they haven't dead. They are not dead. Like, they are still in the upper motor neurons. There's this early stages of symptom onset, and patients have symptoms, but the neurons are still in the brain, but they're unhappy. So once we give NU-9, we make them happy, so then they don't degenerate. So then they stay at that level, and then we stop the degeneration and we improve their health. But then once the neuron is dead, and then there's astrogliosis, microgliosis, and they chew them apart, then it's very hard to bring it back to life, right? So that's why I think treatment as early as symptom onset would be very important. And we haven't done the experiment with, let's say, P90 administration, where neurons actually, let's say, started apoptosis or started their own death, right? And if in that case we can still reverse them back, that will be also remarkable. But we haven't done that study in this particular publication. But thank you for the question. Yes, it's important to make that distinction.
Okay.
So
Anyway, keep going. You were doing.
Yeah, that's perfect. So the other thing that NU-9 was able to do, as Dr. Silverman said, reversed the protein aggregation, or reduced, let's say, not reversed, but reduced protein aggregation. So there was less and less protein aggregation in diseased neurons. So this is also very important.
On the protein aggregation, am I saying it properly that by having this protein aggregation and misfolding of proteins, that the proteins aren't able to recycle themselves and therefore become a problem rather than a building block?
That's right. That's right. Because problems cannot, protein, like ER is the site where proteins are made. So one of the definition of ER stress is because they either cannot fold proteins properly, and proteins may accumulate in ER. So if there's a problem with protein production, protein processing, protein folding, then ER has stress. So ER stress is related to protein problems. So by improving the health of ER, by also reducing the aggregation of proteins, that gives a huge relief to the motor neuron. And so finally, I don't have to deal with this. So we actually we give them a big relief. All right, so then this was only in the SOD1 mice. And Dr. Silverman also made the discoveries of NU-9 based on the SOD1 model. Then we took a very big leap and said, okay, if NU-9 improves the health of mitochondria and ER, and these are exactly the same problems we see in the TDP pathology, right? Would NU-9 also improve the health of upper motor neurons that become diseased due to TDP? The answer could have been no. The answer, we didn't know, but we started the experiments by using the transgenic mice, which was crossed to the GFP, the TDP model, which was crossed to the GFP. So we used this model, and we previously published that this actually recapitulates the pathology in human. It was actually published by Mukesh Gautam, and he's the Along Swim Fellow. So Mukesh has done an excellent job on this paper, and hopefully we will interview him as well. So then the problem, you remember I showed you the apical dendrite problem and cellular degeneration problem in the SOD1? The same problems were also present in the TDP, and most importantly, NU-9 was effective not only in SOD1, but also in TDP. So that was very important, because then if you cover SOD and TDP, you basically cover a huge spectrum of ALS, FTLD. And by improving the health of the upper motor neurons, you also help potentially the HSP, PLS patients, and the upper motor neuron disease population. Then we looked at behavior, right? Does the behavior improve in any way? It's very unlikely that when you improve the health of the upper motor neuron, the behavior improves in the mice. But yes, there was very distinct weight differences, that these were really diseased mice. And the rotarot did not really show much, and that's actually an example of a broad motor function defects. But the latency to fall from a hanging wire showed very important results. And this behavior test was also published by others to reveal the connectivity and the health of upper motor neurons. So this finding suggests that the upper motor neurons are also connected within the upper motor neuron circuit, or within the motor neuron circuitry. So that's very good. This is for the SOD1, and we also received similar results with the TDP. And that's why I think NU-9 is now, is the only compound that improves the health of upper motor neurons that are diseased in two different disease mechanisms. And in a nutshell, because we have a target, and the targets are the mitochondrial defects and ER defects, And we also have a cell-based approach. And these cells, these neurons are important for many neurodegenerative diseases, including HSP, PLS, ALS, ALS-FTLD, and even some patients with spinal cord injury. So if we can improve the health of the upper motor neurons, this would have huge implications for a broad perspective for many patients. And being able to improve the health of upper motor neurons that become diseased due to two distinct causes also, I think, makes this study very intriguing and worth pursuing for the future.
Well,
thank you so much.
That's terrific, Hande, and so it gives everybody in the ALS and PLS HSP worlds really a reason to be optimistic and so forth. I'm curious. I'm going to switch gears a little bit because, of course, whenever anybody hears that there is a compound that has some promise, they wonder where the drug is. And obviously, there are a lot of steps between here and even to get to phase 1 trials. And Rick, I know that you have been through those jumps many times before. One of the things that I left out, am I saying this right, Rick? You've got near to 100 patents and almost 400 articles to go along with your books. Clearly drug development is your wheelhouse. So talk to us a little bit about how we get from here to there.
Yes, unfortunately, we're not ready for humans today. We've shown efficacy, that it does do what we were hoping it would do. And we've done preliminary toxicology on mice and shown that it's actually quite safe, certainly safe at the levels Dr. Ozdinler was using in her studies. But in order to get past the FDA, you then have to do extended toxicology studies under very special conditions. And you have to do it not only in rodents like mice, but also larger animals, dogs or monkeys, which are a little closer to humans. And so we need to do those studies. And then you also have to make the drug that is pure, sufficiently pure for humans. It's okay to use highly laboratory purified material in animals. In fact, the compound we gave to Dr. Ozdinler was greater than 99% pure. But the FDA wants to know if there are any impurities, what are they? And how safe are the impurities? And a whole long list of different things they want. And so you have to have that material made. And so that, in addition to other studies that the FDA would require, this is probably a year and a half, 2 years away from potential approval by the FDA.
Approval for phase 1 trials or?
For phase 1 clinical trials.
Okay.
Yeah.
Okay, because all the toxicity and so forth are the jumps between here and there. And of course, it would be remiss not to talk about the fact that getting a compound through that process is incredibly expensive. What is the, as you get into phase 1 trials, of course, the big expense really starts. What does it take to get from here to the phase 1?
Well, you have to carry out what is known as IND enabling studies, because you have to write what is known as an investigational new drug application, or IND, in order to apply to the FDA for approval to begin clinical trials. The IND includes everything that we've done with the drug. And to get through this IND stage is probably going to cost in the range of $2 to $3 million.
Okay. what do you see or can you foresee as being potential snags or potholes that could come along? I just from everything that Hande was describing and everything that you've described, starting with 50,000 compounds and coming down to effectively one, it's already been so selective. And from the standpoint of the layperson, just almost a miracle what this stuff can do. Where do you see the real potential forks in the road, Rick?
Well, it doesn't seem like there should be, considering we use fairly high dose in Dr. Ozdinler's studies. She's exceedingly careful in what she does and has looked at cells in humans and in mice. As I say, we've shown low toxicity, at least initially, but we've only looked out as far as 1 week. You'll then have to look at toxicity for at least a month, if not more. And so that's where, who knows, something could happen. But in Dr. Ozdinler's study, she was administering this large amount for 60 days. And she hasn't found any toxicities. So I'm feeling confident that this should be able to move along once we get these studies done.
Boy, that's terrific. That's terrific.
So, Dr. Silverman is correct, because when we gave 100 milligrams per kilogram per day, we have given 60 days to all these mice, right? And none of them had any carcinoma, tumor production, or death, or, we did not observe anything abnormal in those mice. So we don't think that there is an obvious toxicity even at the high dose of 100 or, the 100 milligram dose. But maybe they will try different doses. They may want to do a survival assay. They may want to do, overdose and different doses, because once this is in the clinic, the goal is that the patients will be taking them for a long time, right? It's not just one-time delivery. It will be multiple times delivery during their lifetime. And if that's the case, we really need to know, the dosage, and the toxicity of the dosage is very important to identify. And
well, and another bit of optimism, I think, is that as you have identified so many times, the difference between mouse physiology and human physiology, at least when it comes to the upper motor neurons, that they're so similar. And again, for the layperson, that's a source of real optimism.
Yes, that's exactly what I believe, because at the cellular level, all cells need mitochondria to make energy. It doesn't matter if it's a mouse cell, if it doesn't matter if it's a human cell, it's whichever cell, eukaryotic, that requires mitochondria to make energy. And mitochondrial defects are observed in many within the context of degeneration. So if we can actually improve their health with NU-9, that at a cellular level, in my opinion, should be translational.
Yes, the CLP is a very interesting collaboration between the medical school and the Department of Chemistry up in Evanston. And Rick, you're kind of a father of the CLP as well. Do you want to talk just for a minute about chemistry and life processes?
Yeah. Yes. So CLP, that's Chemistry of Life Processes. This was an institute that was started over 10 years ago to connect faculty at Northwestern, both in the Evanston campus and the Chicago campus. That's the medical school and the Chicago campus. And it's all departments, not just chemistry. And it's any faculty that is working in the areas of therapeutics or diagnostics, and from any angle. And it's a way of connecting faculty so that you're aware of other faculty working in similar projects so that you can then collaborate. And we have quite a huge number of collaborations now among CLP faculty. And they occur because of this connectivity that is promoted at Northwestern for the whole endeavor of therapeutics and diagnostics.
Boy, that's terrific. That's terrific. And what a great platform for everyone like Hande and others to be able to have a forum with which to share all that information. It's just terrific. Just terrific. Okay. Well, Rick and Hande, thank you so much for joining us today. We're obviously optimistic about the next steps on NU-9. We're thrilled to be on the team and really pledge our support going forward. So
Great.
Thank you again.
Thank you, Doug.
Thank you so much. Thank you for the invitation. Thank you.